Welcome back to this next video, and this is the part 2 on the protein microarray. A quick recap of the part 1. In the first part of the video, I've told you about the microarray, and then we talked about the protein microarray or the protein chip. And one of the major advantage of the protein microarray was that it was actually screening a large number of proteins in parallel. Then we talk about the different kind of the probes that you can immobilize on the chip, the antibodies, the antigen, the aptamers, the Fe bodies, the full-length proteins or their domains. And then we talked about the sources of the probes that you can actually immobilize on the chip, and the sources were the cell lysate, the recombinant DNA technology, the synthetic peptides, and then the cell-free translation system. And in the cell-free translation system, we discussed about two of the most commonly used cell-free translation system, including the nucleic acid programmable protein array, and then we discussed about the drawbacks of the NAPPA, and then the protein in situ array. Then we talked about the protein immobilization on the solid support and the properties of the solid support, that the solid support, they should immobilize the protein, but they should preserve the 3D structure of the protein, and they should not change the chemical nature of the protein. And then we discussed about the microarray, that these microarray, they are providing a buffered aqueous environment to the proteins, and it was responsible for preventing the cross contamination between samples. And then we discussed about the detection system. Now in this particular video, I want to focus on the types of the protein microarray. And the protein microarray, they are broadly divided into two types. The first type of the protein microarray, they are known as the analytical microarrays, and they are also known as the capture assays. So when you talk about the analytical microarrays or the capture arrays, they uses a library of the antibodies, the aptamers of the Fe bodies that are arrayed on the support surface. So in this particular analytical microarray, the probes that you are immobilizing, they can include the antibodies, the aptamers, or the Fe bodies. Now these antibodies, aptamers, or Fe bodies, they're used as capture molecule, since each binds specifically to a particular protein. Hence they can tell you about the presence or absence of a particular protein in your sample. Now this array is probed with a complex protein solution such as a cell lysate, and this type of microarray is especially useful in comparing protein expression in different situations or in different solutions. For example, the response of cells to a particular factor, for example a drug, it can be identified by comparing the lysates of the cells treated with specific substances, in this particular case the drugs, or grown under certain conditions with the lysate of the control cells. So you can actually compare, say for example, if you have got a population of cells, you treat them with a particular drug, or you provide them with a particular condition, and then you are actually checking the cell lysates in comparison to the control cells, that what kind of changes they are occurring in the proteome of the treated cells. Now another important application, that is the identification and profiling of the disease tissues. Now this particular image is actually showing you how the analytical microarrays, they are getting their desired results. So for example, if this is your sample that you have treated with a particular drug, and this is the control sample, that means both of them, they are the same samples, but one sample that has been treated with a particular drug, and the other sample have not been treated with that particular drug. So this will be your control, and this is your test sample. Now what you do is, when you apply the drug over here, you are going to isolate the proteins or antigens, for example, from the control as well as from your sample. And the next step that you are going to do is, you are going to label them. Now in this particular case, the labeling is done with different fluorescent molecules. For example, if you look at the control one, here the proteins or the antigens, they have been labeled with a green fluorescent molecule. The different kind of the proteins you can see over here, they have been represented with different colors, but all of them they have been labeled with a green fluorescent molecule. Now in case of the sample, all of them they have been labeled with a red fluorescent molecule. So you can actually differentiate between the proteins of the two, the proteins of the control and the sample, by observing the specific fluorescence. If you are getting a red fluorescence, that means you are actually observing the proteins that is present in the sample, and if you are getting a green fluorescence, that means you are actually getting the results of the proteins in the control. But if you are getting a different color, I'll talk about that in a while. In this, if this is your chip and you have these antibodies present on the chip, for example, there are 5 types of the antibodies. This is antibody number 1, this is number 2, 3, 4 and 5. Then you are going to add the mixture of the proteins that you have labeled to this particular protein chip. That means the proteins of the control as well as the protein of the sample, they are now going to interact with these antibodies. Then when you go for the hybridization, and they actually go and bind to their antibodies, their specific antibodies, then what you do is that you go for the detection. The first thing you do is that you apply a red laser, and the red laser gives you fluorescence in all of those particular spots where the red fluorescent molecule that is present. You save that particular image, then you apply a green laser, and the green laser is going to give you fluorescence in all those particular spots where the green fluorescent molecules that have been bound. So the computer that saves both of these images, the images from the red laser, the images from the green laser, and then it is going to merge them. So what you do is you are going for the image capturing and then the analysis. Now if you analyze this particular chip based on the fluorescence, you can see that on the antibody number 1 or on the position number 1, you are only getting the red fluorescence. What this means is that these particular proteins, they are only expressed in the sample condition. They are not expressed in the control condition. What this means is that this is the differential protein that have been expressed only in the sample when that have been exposed to that particular drug, and that has not been expressed in the control. If you talk about the second one, here you are only getting the green fluorescence. That means that these proteins, they are only expressed in the control and not in the sample. If you talk about the this orange one over here, you can see over here that the red fluorescent molecule, they are present, the green fluorescent molecule that are present, but the red fluorescent molecule, their number is high, or their fluorescence is high as compared to the green one. This means that during this particular condition, when you apply a drug to this particular sample, the expression of this particular gene actually will be high, and the expression of the this particular gene in normal condition, that will be low. So it is actually showing you a comparative expression of this particular protein. If you talk about this one, they are equally expressed, these particular antibodies. These antibodies, they are not expressed in any of the control or any of the sample. And here you can see that this one is more expressed in the control condition as compared to the treated condition. What I mean by this is that you are actually going to see the differentially regulated protein, that which kind of the proteins they are expressed in the sample condition, in the treated condition, and which of the proteins they are actually expressed in the control condition. So the analytical protein microarray can actually show you the differential expression of the proteins under the control and the treated condition. The second type of the protein microarray, that is known as the functional protein microarray. Now the functional protein microarray, that is also known as the target protein arrays, and they are constructed by immobilizing large number of purified proteins. So this is different than the analytical microarray, where you are actually immobilizing the antibodies, the aptamers, or the antigen. Here you are actually immobilizing a large number of the purified proteins, and the goal in the functional protein microarray is to identify protein-protein, the protein DNA, the protein RNA, protein phospholipid, and protein small molecule interactions. So here you are actually focusing on the functional aspect of the protein. In the analytical microarray, you are actually focusing on the differential expression or the presence or absence of a particular protein under different conditions. But in the functional microarray, you are actually focusing on the interaction of the purified proteins with the protein, DNA, RNA, the phospholipid, or any other small molecules. The functional protein microarray is also used to assay the enzymatic activity or to detect antibodies and demonstrate their specificity. Now, the functional protein microarray, they actually differ from the analytical microarray, is that the functional protein microarray are composed of containing full-length functional proteins or protein domain, because here in the functional protein microarray, you are actually interested in the functional aspect or the interaction aspect of the protein microarray and hence their activities. Now, these protein chips, they are used to study the biochemical activities of the entire proteome in a single experiment, because in the first part of the video I've told you that the major advantage of using the protein microarray is that you can actually study the entire proteome, or you can study a lot of proteins in a single experiment. So these are the two major types of the protein microarray, the analytical one and the functional protein microarray, and both of them, they have got their specialized functions. Now, if you talk about the functional protein microarray, if this is your sample, what you do is that you are going for the extraction of the proteins, just like the previous one. Then you are going to label them. As in this particular case, you are only focusing on a single sample, so you can use a single type of the fluorescent molecule. In this particular case, the green one. So you have labeled all of the proteins. Now, on your protein chip, you have immobilized the full-length proteins to whom the interaction of which you are interested. For example, if you are interested in the you want to see that which of your proteins in your sample are going to interact with this particular protein, or with this particular protein, or with this particular protein, because you already know on the chip that on this particular position which kind of the protein is present, on this particular position which kind of the protein is present. So when you hybridize your labeled proteins on the protein chip, you can actually go and see that which kind of the proteins they are interacting with this particular protein. So as you can see over here, from this particular lysate, this protein, this one, this protein, and this protein, they are actually interacting with these specialized proteins, and you can actually say that in your sample there are proteins present which can interact with these proteins but not with these kind of the proteins, because if there is no protein interaction, that means you are not going to get any fluorescence on these spots. So on this particular spot and on this particular spot there will be no fluorescence, but on this particular spot, as you are getting the fluorescence, that means that interaction is taking place over there. So if you can see over here, you are getting these black spots over here which indicate that there is no fluorescence, and you are actually getting the fluorescence on these particular spots. Now, if you talk about the applications of the protein microarray, now the functional protein microarray, they provide a very flexible platform that can characterize a wide range of the biochemical properties of the spotted proteins. As I've told you that these assays, the functional protein microarray assays, they have been successfully developed to detect various types of the protein binding properties. For example, the protein, the protein DNA, the protein RNA, the protein lipid, the protein drug, and the protein glycan interactions. And you can also identify the substrate of various classes of enzymes, such as the protein kinases, the ubiquitin E3 ligases, and the acetyl transferases. So these are some of the major functions of the functional protein microarray, major applications of the protein microarray that have been so far achieved. Now, another important application of the protein microarray is the identification of the biomarkers. And these biomarkers, they are very crucial tool in the expeditious detection of infection or diagnosis of the autoimmune disease. So protein microarray can actually help you in the identification of the biomarkers, thereby helping you in the diagnosis and hence the treatment of the different diseases. Another important application of the protein microarray is the identification of the protein post-translational modifications. Now these post-translational modifications, they are very important as far as the structure and function of the proteins that is concerned. And among the 100 of protein post-translational modification that have been identified with the help of the protein microarray, that include the glycosylation, the enzyme-dependent reversible protein phosphorylation and dephosphorylation, ubiquitination and deubiquitination, and the acetylation and the deacetylation. And perhaps these are the most well-studied post-translational modifications with the help of the protein microarray. Now, a new trend in the field of the host-pathogen interaction is to use the host protein microarrays to survey the relationship between the pathogenic factor of interest and the host proteome. Because this kind of the host pathogen interaction, that is very important, especially in the host virus interactions, because after the entering the host cell, the viral genome and proteins, they are in direct physical contact with the host components, whereby a pathogen can hijack or exploit the host pathways and machineries for its own replication. So if you know the host pathogen interaction, and if you identify the target of a particular pathogen that it is targeting in the host, and if you can block that particular pathogen protein by the use of some kind of a drug, you can actually stop the host pathogen interaction and hence the disease. Now there are a lot of challenges as far as the protein microarray is concerned, and despite the considerable investment made by several companies, the protein chip has yet to flood the market. Now the manufacturers have found that protein microarray, they are actually quite difficult to handle in comparison to the DNA microarray, and a protein chip that requires a lot of more steps in its creation than does a DNA chip. The major challenges that the protein microarray it faces in the current era, that include finding a surface and a method of attachment that allow the protein to maintain their secondary or tertiary structure, and thus their biological activity and their interaction with other molecules. Because if the surfaces, they are immobilizing the protein, but at the same time they are changing their secondary or tertiary structure, they will not be able to identify their targets in your test samples. So this is one of the great challenges in the protein microarray, to find a surface that is suitable for the protein microarray. Another important challenge that a protein microarray face in these days is producing an array with a long shelf life, so that proteins on the chip do not denature over short time. Because these proteins, they are very sensitive to their environment, therefore they have got very short shelf life. So one of the challenges is to make the protein array with a long shelf life, so that they do not denature over a short period of time. Another important challenge is identifying and isolating antibodies or other capture molecule against every proteins in the human genome, because currently you are not able to study the whole proteome of the human being utilizing the protein microarray, because of the unavailability of the antibodies or capture molecule against every protein in the human genome. Another challenge is that during the protein microarray analysis, the quantifying the levels of the bound proteins while ensuring sensitivity and avoiding background noise. The synthesis of the protein and their extraction in the pure form, that is a very laborsome process, and once you immobilize them on the chip and you go for a particular detection, those proteins usually denature and you cannot reuse them. So one of the challenge is that you make such kind of the protein array so that you can extract the detected proteins from the chip in order to further analyze it, or you can use it in some other kind of experiments. Currently you cannot do this. Another challenge is reducing the non-specific binding by the capture agents, And another important challenge is the capacity of the chip must be sufficient to allow as complete representation of the proteome to be visualized as possible, because one of the problem with the protein microarray, especially their detection system, is that the abundant proteins, they usually overwhelm the detection of the less abundant proteins, such as the signaling molecule and receptor, which are generally of more therapeutic interest. Say for example, if a protein is expressing in a very high quantity and is comparison to that a protein that's expressing in a very low quantity, so usually these low quantity proteins, they are not observed by the available detection system. So one of the major challenge is to make such detection system that can tell you about the expression of the abundant as well as the low abundant proteins. So if you like the video, please subscribe to my channel, share it with your friends, and hit the like button.